Hello, and welcome to this presentation on the anatomy and physiology of the equine immune system. Today we'll explore the intricacies of the system, including its organs, cells, mechanisms, and how they all work together to protect the horse from infections and diseases. To start off, I'm going to talk about the main structures and components of the equine immune system. The most important tissues are the bone marrow, the spleen, the thymus, the tonsils, the lymph nodes, the mucous membranes, the skin, and the vessels or the blood vessels. These structures all work together to filter and remove foreign substances from the body to activate and coordinate the immune system response. For starters, the bone marrow in horses contain pluripotent stem cells which divide into myeloid stem cells and lymphoid stem cells. Myeloid stem cells give rise to granulocytes such as neutrophils, eosinophils, basophils, while the lymphoid stem cells will produce areuginocytes, specifically lymphocytes. Neutrophils are the most common white blood cell in the horse and are the first line of defense for the host immune system. They rapidly move to sites of infection and inflammation. Eosinophils are associated with parasitic disease and allergic conditions, while basophils are responsible for the onset of infectious, allergic, and autoimmune disease. Mast cells are involved in the initiation and preservation of a number of inflammatory conditions. Finally, lymphocytes play a crucial role in antigen recognition, antibody production, cytotoxic attack on infected cells, immunologic memory, and regulation of a specific immune response. To briefly visit blood tests, white blood cell count in horses can indicate various blood disorders. These can include physiological leukocytosis, lymphocytosis, neutrophilia, neutropenia, eosinopenia, lymphopenia, and monocytosis. An increase in monocyte numbers may indicate bacterial infection, chronic inflammation, or stress, while an increase in neutrophil numbers is often due to bacterial or viral infection, injury, stress, or drug administration. Basophils are rarely found in blood samples, however, but their presence may indicate a long-standing allergic disease or ongoing recovery from colic. Foals normally have more lymphocytes than adults, and lymphopenia is commonly caused by corticosteroids. The next major organ that aids in the immune system is the thymus. It is a specialized organ of the immune system located in the chest of the horse. The thymus plays a crucial role in the development and maturation of T lymphocytes, which are important immune system cells that help protect the body against infection and disease. As the horse ages, the thymus will actually decrease in size and activity, but it will still remain an important site for immune system development. And lastly, the final organ I want to talk to you guys about is the spleen. The spleen plays an important part in both the immune system and the circulatory system. It contains a high reserve of immune cells that can be quickly mobilized to respond to an infection or an injury. The spleen can do this by contracting and releasing stored red blood cells into circulation in response to physical activity or stress, which can help to maintain the adequate blood flow and oxygenation to the tissues of the body. The development of the immune system. The development of immunity in horses from foal to adulthood is complex, with some innate immune responses being functional from birth and others developing slowly over the first year of life. Interferon gamma production starts slowly after birth, which explains the increased risk of foals to certain pathogens. The delay of Th cell development and Th2 immunity during the first few months of life also explain the reduced responsiveness to traditional vaccines. The thymus plays a key role in the development and the maturation of T lymphocytes, which are used to protect the body from infection and disease. Once activated, these lymphocytes can move to lymphoid tissue where they help to mount an effective immune response. The nonspecific immune system. The immune system has two main components, the innate immune system and the adaptive immune system. The nonspecific immune system, which we'll be focusing on, is also called the innate immune system. This includes physical barriers like skin and mucous membranes, effector cells such as neutrophils, eosinophils, basophils, and macrophages, and soluble mediators such as cytokines and antimicrobial peptides. Allergic hypersensitivity involves white blood cells releasing granules that cause allergic symptoms when the body recognizes an allergen. Cytokines are proteins that regulate cell response during inflammation and help increase local circulation. The complement cascade enhances the abilities of antibodies and phagocytic cells to clear microbes and damaged cells from the body. This complement system is a plasma protein cascade that can be activated directly or indirectly by pathogens, which will lead to various effector functions. The specific immune system. Before jumping into the adaptive immune system, I'm going to briefly touch on the specific cells in the adaptive immune system. There are several different types of T lymphocytes, including cytotoxic T cells, helper T cells, memory T lymphocytes, and natural killer T lymphocytes, each with specific roles in the immune system. Cytotoxic T cells directly destroy infected or abnormal cells, while helper T cells coordinate and regulate the immune response. Memory T lymphocytes remember previous infections, and natural killer T lymphocytes are part of the innate immune system that can directly attack and destroy infected host cells. The memory T cells, in the case of a reinfection, will produce immunoglobulins or antibodies. These proteins are produced by B cells and are designed to recognize and neutralize specific pathogens. There are several types of immunoglobulins in the horse, including immunoglobulin A, M, E, and D, and 7 subclasses of the immunoglobulin G. Immunoglobulins play a crucial role in protecting a horse against infections and diseases. Moving forward to the adaptive immune system as a whole, it is highly specific to the pathogen that induced its activity. It involves antigen-specific immunity, and it is carried out by the white blood cells called lymphocytes, which include B cells and T cells. B cells secrete antibodies that bind to foreign antigens, which inactivate them, while T cells react directly on foreign antigens present on surfaces of infected host cells, killing them before the virus has a chance to replicate. The clonal selection explains the functions of cells of the immune system in response to specific antigens. The activated B cells differentiate into plasma cells that produce a vast amount of specific antibodies. Additionally, there are natural killer cells that kill virally infected cells through a non-specific manner. These killer cells are activated by the immune system and attach to an infected cell or the target cell by secreting perforins and granzymes that induce cell death to eliminate viruses and tumor cells. MCH, or major histocompatibility complex molecules, play an important role in the immune system by aiding in the identification and destruction of infected or abnormal cells overtaken by pathogens. MHC-1 molecules are expressed on almost every nucleated cell in the body except red blood cells, and are present in fragments of cytosolic and nuclear origin of the cell surface. MCH2 molecules, on the other hand, are expressed on antigen-presenting cells and present antigens derived externally from the cell, such as from bacteria to the helper T cells. Activated helper T cells secrete cytokines and other types of signal molecules which stimulate and coordinate an immune response to a threat. In conclusion, the equine immune system is a complex and sophisticated network of organs, cells, and mechanisms that work together to protect the horse from infection and diseases. The thymus, spleen, lymphoid tissue, and immunoglobulins are all essential components of this system, each with a unique role in the immune response. MHC molecules aid in the identification and destruction of infected or abnormal cells overtaken by pathogens, while T lymphocytes play a crucial role in coordinating and regulating the immune response. With this knowledge, we can better understand and appreciate the amazing abilities of the equine immune system. Thank you so much for watching, and I hope you learned something.